So this first round of questions, the panelists will be talking about the focus of their project. So what is the focus of your project? How did your project get started? And what are you trying to accomplish? What kind of progress have you made so far? You want to start?
Yeah.
Yeah.
Hi everyone, my name's Marissa and I am here representing the Georgetown Shellfish Committee. So we were working on a project that was basically an effort to diversify Georgetown's shellfish resources. I do have a slide that they're getting up right now, just to orient everyone to where Georgetown is, first of all. On the coast, we're in the mid-coast area. So yeah, that red circle up there on the map is Georgetown. So Georgetown, like many other coastal communities, is suffering from the decline of the softshell clam resource. Unlike a lot of other coastal communities in our area, that's really the only resource that shellfish harvesters have. We don't have a population of quahogs. There's not really an active razor clam industry, so it's really just the softshell clam population, that's it. So these harvesters are really struggling in recent years because there's been such a decline in the population. So this was a project that was really focused on trying to diversify those resources by transplanting quahogs into Georgetown. This was a project that went over a 2-year time period. We started in 2018 with town funds, and then in 2019 we were fortunate to get some broad reach funding to continue the work. So that map on the right-hand side is Robin Hood Cove. That's the area that we transplanted the quahogs into. It's about 3 miles long from the mouth to the head of the cove. Actually, it's probably more like 3.5 miles long. It's very protected in terms of water currents and wave exposure and things like that. And this was the area that the harvesters really wanted to focus on for this transplant project. So if you advance, oh, perfect. In 2018, we transplanted 10,000 adult quahogs into the cove, and then in 2019, we transplanted 40,000. There's a conservation closure currently on the harvest of all quahogs in Georgetown, and that's really meant to allow this population that's been transplanted to have time to settle and adjust, and so there's no harvesting of these quahogs. The idea here is that hopefully this is going to create a population that one day they can actually harvest. And to give you a sense, that cove, that Robin Hood Cove area, even if the larvae that the quahogs might be eventually breeding and producing, even if it leaves the cove, it would have to go around a huge piece of Georgetown's coast to actually leave that area. So the hope is that, you know, there could be some self-seeding of this population within Georgetown and the surrounding flats, even outside of this cove. So if you want to go to the next slide. So this is just some of the results. So there's the sites that we planted at across the top in these tables, and then the number of quahogs planted at each site, the percent recovery. So it's really hard to get at survival of something that you put in the mud that can move. So we had plots that were very clearly marked where we planted a known number of quahogs, and we went back several months later and we harvested those plots to get a sense of how many were still there and how many survived. So you can see, you know, it ranges from 17% to 56% in 2018, and then it's right around 40% at the sites in 2019. It's hard to say if this is reflecting survival. You know, we were harvesting these plots and we didn't really see a lot of empty shells that might indicate that they were dead quahogs. You know, of course they can move outside of the plots, but also these areas were still open to harvesting. Even though there's a conservation closure against harvesting quahogs, there was still active digging of softshell clams and worms, so there was the potential for people to have dug through these plots as well. So it's kind of hard to say exactly what the recovery rate is reflecting, but at least we were able to find that there were healthy individuals inside of all of these plots when we went back. And we also looked at spawning, so we took a subsample, dissected them, and looked at them under a microscope to see if they were actually spawning. So there's, you know, a lot of things that we've learned from this so far, and each one of these is a new. I'm regretting putting all of these animations into this slideshow. So we found that we had greater success with smaller individuals. So the first year that we did this in 2018, we were using really large cohogs. They're like the chowder-sized cohog because they're cheaper. We were, of course, buying all of these cohogs that we used. So the larger the cohog, the less expensive they are, which made a lot of sense for trying to get more and more of them. However, those larger cohogs didn't seem to do quite as well, likely due to some energetic constraints that the larger cohogs were maybe experiencing in the stress of that move. So the second year we used smaller cohogs. That seemed to work better. They were more expensive though, so that's sort of the trade-off there. The condition of the cohogs was very important, and it was a big challenge for us to be able to coordinate buying thousands and thousands of cohogs at a time from a dealer who had to sort of stockpile them in order to fill our order. So oftentimes the cohogs were out of the water and in storage for longer than we would have liked. So that led to sort of varying stages of condition that the cohogs were in before they were planted. So the second year when we did this, we really tried to work much more with the dealers. We actually worked with a couple of different ones, but primarily Ipswich Shellfish, who was really good about trying to make sure that the cohogs that we were buying had only been out of the water for 3 to 4 days at most, to sort of minimize stress. If you want to, one more, yep. Of course, the survival or recovery rate was difficult to measure, which I had mentioned before. And from here, it's really about long-term monitoring for us. So we're going to be, trying to measure the impact of having transplanted 50,000 adult cohogs into this cove. We'll be using recruitment boxes within the cove, but also elsewhere in Georgetown, to try and, see if we do see any sort of larval settlement. We're also working with Gabby to do some drifter studies within the cove to see how the water is actually moving. So one of the reasons that the clam harvesters chose this cove is because they dig in this cove all the time and they see that things tend to get stuck in the cove, right? So there's a lot of things that never leave that cove. So for instance, oyster cages that break free and are floating tend to get stuck in certain places in the cove. So they think that there's, some water movement within that cove that might actually lead to some of that sort of larval retention in that area. So we want to be looking at sort of the water movement with these drifter studies. And then I think moving forward, there's a lot of sort of work you can put into trying to rear cohogs from seed that could be actually bought at the seed stage and used for these sort of conservation and seeding measures rather than adult cohogs. And I think that's sort of where we would be headed next as well for any additional efforts to introduce cohogs into this area.
There are Stonington. The two towns, we are a joint committee, and that's where we are. In 2018, we produced 600,357 clams, which between the two towns, and that was 9% of the clams landed in Maine, which makes it a very important industry. In 2018, there was 62 diggers doing that. Today we have like 82 or so. At any rate, it's a very important industry for us. And we had a problem in Long Cove where it came in and out of being closed. And we walked around and we hadn't found anything that we could pinpoint. We're looking for, the obvious, some septic system that was failing or something. And the same was true. Long Cove was gone in and out. Deep Hole was somewhat new. And people are upset when things close, they get upset, especially if that's where they like to go. So when it came opportunity to do microbial tracking, we jumped at the chance, and when the grant came up, so that's what we did. So we want to get to our next slide. All right, we did a number of samples. We tried to do it every 2 weeks. Over the course, we started, I think it started like June 10th, and we went to October 31st. Out of that, we managed, the water quality was much better than we'd expected, which is good, except when you try to find, bacteria to study. So at any rate, we managed to get 7 that we sent down to New Hampshire, Steve Jones, and he did the testing there, and what we came up with was it wasn't a human source. It was birds, dogs, and mammals, I guess, are always in a study. So they don't really count because anywhere in Maine you're going to find mammals. So our main problem seems to be birds and dogs, which are hard to do. We originally thought in Crockett Cove, that it would be beaver. Beaver didn't really show up as a problem. What showed up as a problem was dogs, which probably were coyotes, because there's no real, I don't really see people letting dogs out there. It's more of a wild area. But there's a pond, if we can go to a deep hole. The other map along Cove here. Deep hole, up in Deep Hole, where we took a sampling, there was a pond coming out that ran into the cove, and we thought, well, that's probably a problem, which it was. It wasn't as bad as we thought, but it was a problem, and that turned out to be mostly birds. So that's where we are. We checked it. We got samples back, and we have you don't have to have the graph here.
No, I don't.
Okay, that's fine. At any rate, we do have a graph in our study if you wanted to. I'm sure you could get a copy. But basically, Long Cove is back open. It was closed. It's open now. We kind of think it'll probably stay open. Deep Hole closes in July. It opens back up in October. And in Deep Hole, our highest reading was in October 31st, which was a little odd because the water temperature was cold. In Crockett, Long Cove was mostly pretty good. But most of our problem seemed to be deer and dog. We're going to try to inform people around Long Cove, particularly Long Cove. There's a park up where, up at the other end of Long Cove, between the two sites. There's the Meredith Park, and people walk their dog there, and that may be part of the problem. There are signs that say you're supposed to clean up your dog. I think we'll probably do a little more informing of the general public of how important it is to keep the dogs off of the clam flats or anywhere near it. Basically, that's it. I would like to thank everyone in Lemoyne, DMR, because they did training for us to show us how to take samples. The lab was wonderful, and when we went up there, I went up there every 2 weeks, and they were good to work with, and they were friendly and very helpful as well as the people that took samples. So, and of course, DMR will continue to take samples at their normal sites. I think that's about all I have to say about it. Thank you.
I'm the chairman for the St. George Shellfish Committee, which consists of 5 towns, of Warren, Thompson, South Thompson, St. George, and Cushing. I picked my sites off the state pollution site map, because we have 5,980 acres under some kind of state management. This is including conditional areas and restricted and all that. So I wanted to be, when I talk about an area, I wanted to be on the same page. So Jeffrey Schiff was kind enough to download everything into a handheld GPS for me. So we went to work. It was the grant money we got from Brady, and started doing some DA testing. We was lucky enough to get 27 tests in this summer. I had some human come back in the Site 29, which is actually, I guess you can't see the sites on there. They have dots, and the little dots have like St. George, and one side of St. George is all WU, and South Thomaston and everything is BW, with numbers beside them. So we went to work, and we had some up in the particular area we call the Bay, We had a human site come back as human. I started working with a wastewater treatment plant. Actually, my advice there, if you just go right at the top, start with the selectmen, or whoever at the head and work down, because if you start at the bottom, they kind of put you off, drag you around a little bit, you don't get nothing done. So go right to the top. Start at the top, work way down. But before we had this DNA, we had a little key steps in for looking for human waste. It's just a sponge that was developed by Fran, who had before Hannah, who worked for the state. It was a sponge and a black light. It soaked up like for human. Hopefully, it was looking like the dish soap and all that, and that would give us a clue that we had a leak in leach field because we were looking for phosphates. This is in the soap product. And so anyway, I had out of the 27 sites, I had 2 humans, 7 inclusive, 1 dog, 9 bird, 2 inclusive for bird, all 27 sites tested for mammal. That seems to be our biggest problem. What are we going to do with these wildlife? That's keeping a lot of our areas closed. I don't have an answer. I'm hoping someone half a lot smarter than done some science will be able to tell us. It's just, with 5,908 acres, and mammals are keeping it down. That's a lot, because we do have a lot of area. And we could move our sites out, probably get a better quality, because we all know the bacteria grows in fresh water when the summer's warm. The water's warm in the summer, hits the soil, gets out through there, it deteriorates, breaks down. But the state sites, most of them, not all of them, that I have been on, all are in close to shore, all have a brook coming out to them. So there's always fresh water right there being mixed where the sites are. So we're always going to get a mammal test unless we can get them sites moved out. But yeah, like I said, with the sites being closed, and all our digging now is in coastal shore. So I don't know what would happen. If we move them out, we'd just be still, we wouldn't be in got clean water, and drew a line. The inside was still closed, so we're still the button heads. How do we get this under control? I really don't have an answer. If you want to bring the pictures up, we can show. That's me. I worked with the guy in the West Gig River. We took 6 samples over there. He was working, his name was Wes Pratt. He's kind of a semi-retired biologist that moved up this way. And he was very interested, and he was working with DEP last summer. And we worked together on doing this on the West Gig River. That is, every site we've done was by boat at high tides, except for this one site, because they have a bridge, and I can't get underneath the bridge. It's real low up in here, where we walked in and took that last test. You can't see them, but out there it's loaded with oysters all the way down through harvesters. And this area is closed, so they must be decking their oysters, I don't know. But in the Big River, we had 2 bird, all tested, like I said, mammals, and 1 inclusive for human. We got to go back in the summer and we'll check the ones that come back inclusive and make sure, since we can't get a better reading on them. And the only thing we'd like to keep know about, we did that QPC stuff. We paid the extra, had that done. I really need to catch up with either Stephen Jones or someone that knows to break all them numbers down for me. Hopefully, been talking back and forth. We can meet today, get a chance. And basically that's it. What are we gonna do with these animals? I haven't had an answer.
So I work with the town of Yarmouth. I'm on the Shellfish Commission in the town of Yarmouth, and we similarly wanted to look at water quality in the town. And we decided when we wrote the grant to pick the area you can see in the circle because it had the highest P90 score by an order of magnitude of any other score within the town, so we thought that would be a good place to start. And the other thing that we were concerned about was thinking about, thinking about how we maintain data. So one of the problems that we seem to have is that people, collect some data and then it goes away, and then other people collect some data and then it goes away, and then people try to remember what they collected, and it's just based on people's memories and not actual data. So we were trying to think of how would we be able to collect some data that would be able to stay with the town beyond all of our involvement on the Shellfish Commission. And so what we did was, so we were doing both shoreline surveys and water quality testing. So almost everything changed from our grant, from where we thought we were going to be. For instance, we found out after we wrote the grant that our sewer treatment plant was super happy to test all of our samples for us, to give us a first round pass at what were they finding, which of course saved us money for all the samples in which we didn't have to get further tested because there was no actual bacterial contamination, which was great. It did leave us with a little bit of a problem in that they could only test, because they have their normal testing times, so they could only test on Wednesdays, and so that meant that we had to figure out, Wednesday, high tide, and that limits your days. So everything kind of dragged out a little further than we thought it would. But what we developed was an online mapping application so that when we were out collecting data, people took their phones with them and were able to put the data in a data layer that will reside with the town and hopefully continue to be updated. So the things that we were adding to that data layer, so for instance, on this one, you can see that it was just a point on the shoreline survey where they found a drainage pipe coming down between some rocks, and then they took a picture of it, right? So we now have a record, and when we talked to the town, what we found is that the code officer was very interested in being helpful, but he is, of course, super, super busy. And so the more we could narrow things down for him so that he could go to a particular person rather than try to figure out all of the details, that was going to make the difference. But he was perfectly willing, between him and the town engineer, to go and talk to individual homeowners once we had evidence that they could talk to them about. And so this was able to give the town some evidence. So that was one part of it. And then the other part was looking at, from the individual parcel standpoint, being able to walk along, and you can see we haven't filled in all this data yet because we're hoping that's part of our next plan in the spring, but be able to walk in the springtime through and looking for evidence of failed septics on the shoreline above the water. And you can see on that map, it's a little hard to see, but there's like a little purple polygon in the middle. And so the other thing that our next steps are kind of thinking about is that we really want to know where all the septics are physically located, so that when you do that, when you're walking along, if somebody's parked a truck on their leach field, it's not always obvious that that's the leach field because there's a truck parked there. So we want to know if that's the case. And so in order to do that, we need to go through all the plumbing inspector records for the town and digitize those. And so this mapping application is going to allow all that data to be pulled together, and then it'll be a permanent record for the community to have, so that as we move forward in trying to identify the location of areas where we have issues, we can both show evidence that we've cleaned things up and have evidence of where we still need to do more work. And that was sort of the goal of the project.